Good afternoon. Welcome to our research symposium titled The Future of the Brain Initiative. It's an exciting time at an exciting conference to discuss recent advances in neuroscience and the future of the brain science in the context of the Brain Initiative. My name is Xiangmin Xu. I'm a professor of anatomy neurobiology and director of the Center for Neurocircuit Mapping at the UC Irvine. I'm the moderator of today's symposium. I'm very pleased that we have three distinguished speakers for our symposium. Dr. Ngai, director of the NIH Brain Initiative, will share the current state and future plans for the Brain Initiative and discuss opportunity and challenges for neuroscientists. Please join me in welcoming Dr. Ngai to our symposium. Dr. Michelle Jones, London, Chief of NIH's Office of Programs to Enhance Neuroscience Workforce Diversity, will discuss her broad experience and perspectives on neuroscience training. Please join me in welcoming Dr. Jones, London, to our symposium. Dr. Xiaowei Zhuang, HHMI investigator and professor at Harvard University, will talk about her exciting research and will discuss her experience opportunities for technology transfer and commercialization. Please join me in welcoming Dr. Zhuang to our symposium. So for this symposium, each speaker will give a 30-minute talk in the order as shown in the slide. And at the end of all three talks, then there will be 20 minutes for question and answers. So you can feel free to address questions to all the speakers. So with this, I would like to ask Dr. Ngai to come to this podium and start his talk.
So thanks, Xiaoming, and the organizers for inviting us to give this symposium. We're very excited about the Brain Initiative. I hope that you will be excited about hearing what we're up to as well. So let's see. We got things going here. Okay. So the mission of the Brain Research through Advancing Innovative Neurotechnologies, or Brain Initiative, is to revolutionize our understanding of the human brain by accelerating the development and application of new and innovative technologies. This initiative was announced by the White House in 2013, with the first awards being made in 2014, and represents a partnership between 5 US federal agencies as well as quite a number of private foundations. Now, the efforts of the NIH Brain Initiative have been guided by 2 strategic plans. The first was the so-called Brain 2025 report that was authored by a group commissioned by then NIH Director Dr. Francis Collins as a working group of the advisory committee to the director, and this group was chaired by Drs. Corey Bargman at Rockefeller University and Bill Newsom at Stanford University. 5 years later, so that report was issued in 2014. 5 years later, another report was commissioned by Dr. Collins, the so-called Brain 2.0 reports, and the neuroscience portion of that report was chaired by Drs. Catherine Dulac and John Mansell. And that represented a look at the progress made over the first 5 years and a look forward to see where the opportunities are and were. And the work I will describe to you today really benefited from these strategic plans. It's really set us on what I hope to convince you is a very productive course. Now, the NIH Brain Initiative, the NIH's portion of the Brain Initiative, represents an initiative that spans across 10 of the 27 NIH institutes or centers, or ICs. And we are uniquely situated to perform cross-cutting research that is really beyond the capabilities or even interests of any one NIH IC, but instead actually leverages the capabilities across these ICs for the benefit of the missions across the entire NIH neuroscience mission. And so our goal here is to develop and apply new tools for understanding how neural circuits underlie complex behaviors in both health and disease. Now, to achieve this goal, we feel it's very important to leverage emerging technologies to enable these new discoveries about circuit function, to use these discoveries as a foundation for developing new therapies for devastating human brain disorders, and very, very importantly, to disseminate and democratize these technologies, both for basic discovery as well as for clinical applications, and very importantly, for the benefit of all. Now, I just want to give you a very brief overview of the brain funding history. It was started again in 2014 during the sequester. These are kind of dark times, with a very modest budget in the NIH of $46 million. And as you can see, the budget has grown substantially, where today we are working with a budget of $680 million. And these funds come from two sources. One is are monies that are sent to the 10 brain ICs in part of their base, what we call the base funding. And on top of that, in 2016, Congress passed the 21st Century Cures Act, which adds additional money, which basically has facilitated this large increase in funding. So what have we done with these funds so far? We funded over 1,100 investigators through about 1,000 awards across over 200 institutions that have resulted in well over 6,000 publications through 2022 in a variety of journals, really reflecting the diversity of the kinds of science that we've been interested in funding. Here's just a word cloud that highlights for you some of the areas that we've been covering, neuron, of course, but also model systems as well as humans. And this is just a graph of the productivity of these publications over time. Slight dip in 2022. Have you seen that across NIH? I don't know if it's COVID related or not, but nonetheless, I think if you dig down into the actual papers that have been published, they really have been of quite high impact. So today I'd like to leave you with three key takeaways. The first is that brain-funded advancements in tool and technology development, they're already making their way into the clinic. We are already showing hope for having positive impacts on humans and human health. The second takeaway is that our research teams are developing new resources and technologies that are laying down the foundation for future discoveries and future cures. And then finally, we are creating a new paradigm for doing science that's accelerating the pace of discovery. So what we're really driving at is we want to accelerate the pace of discovery in neuroscience research, both for the understanding of how this remarkable organ works, the human brain, but really ultimately in finding new cures for human brain disorders. So let me just start with this first key takeaway. And what I'll do here throughout is I'll just give you a few examples of each of these claims. Okay. So here's a study that was just very recently published by the Capagrossa group in Pittsburgh. So in cases of stroke or spinal cord injury where the corticospinal tract is damaged and cannot reach effectively the local spinal circuits to affect motor control, the idea here is that the local spinal circuits distal to the lesion are intact. So perhaps that even if the signal's coming down, say, in a stroke or in a partial spinal cord injury, if there's some signal coming down to that local area, if there's a way of enhancing the readout of that, perhaps one can effect some recovery of motor function. And this is exactly what they did here in the case of stroke. So this is a case study with just 2 patients. And what they did is they placed some epidural stimulators over the cervical spine in 2 patients that had stroke that affected the movement of the arm. And what I'll show you next is a video of a patient who was implanted 9 years after her stroke. Let's see. And if you could play the video, please. So they actually quantitated the improvement of motor function through this epidural stimulation of the cervical spinal cord. So here she is. The task actually is to pick up, I think it's a chicken nugget, and eat it. So she had a right side stroke, thalamic stroke. And you can see here with the stimulator turned off, she's having quite a hard time grasping the fork, stabbing the meat, or whatever it is, and bringing it to her mouth. Now, the amazing thing, and you'll see this in a moment with the stimulator turned on, is that the improvement was immediate, suggesting that the circuits were there, they just needed to be amplified in some way to function. The other interesting thing was that 1 month after the study was over, the patient, and there was one other patient as well, showed continuing improvement. So that suggests some plasticity in the circuits as well. So here it is with the stimulator turned on. needs a little bit of help grasping the fork, but then I hope you can see that there's a marked improvement. And again, this improvement happened almost immediately, essentially immediately. There was no other training associated with this therapy. So this is really giving us a lot of hope for helping patients recover function following stroke. The same group has also used the same kind of stimulator for recovering lower extremity function after spinal cord injury. And in those studies, quite interestingly, they were able to map the circuits that were affected following this stimulation, and were able to show plasticity in the circuit and actually map it down to the actual cell types using spatial transcriptomic mapping. So this really portends a great future for functional recovery following stroke or spinal cord injury to re-engage the local spinal circuits in a way that will be informed by an actual understanding of the circuits involved in these processes. So again, this is just a very preliminary case study with 2 patients, but showing great promise for how these kind of technologies actually can already be deployed to help people. So in the second set of studies I want to talk to you about involves the use of deep brain stimulation for personalized treatments for neuropsychiatric disorders. So I'm sure most, if not all of you, are aware of the use of deep brain stimulation for treatment of movement disorders, for example, associated with the symptoms of Parkinson's disease. And now what we're seeing is the application of bidirectional deep brain stimulators, which can actually measure and read out biomarkers for different mood states, and then in a very precise way, stimulate certain regions of the brain to bring the patient into a better state. So here's just a set of pictures from Samir Sheth's group, where they use holographic augmented reality to identify areas in the patient's brain about where to place these DBS leads, and using these leads as well as stereo EEG leads implanted over the course of about a week and a half, they monitored neural activity together with objective as well as subjective readouts of the patient's mood state, and were actually able to design for that patient stimulation parameters that move this patient into a better mood state. So here's just a graph showing clinical score for poor mood state. You can see it consistently falls down over time. That blue bar shows when they, without telling the patient, they turn the stimulator off. And the patient's mood kind of went back to a bad state, and they just turned it back on, and the patient went back into remission. So the key thing here is that when we're thinking about issues around treatment refractory depression, OCD, binge eating disorder, we know generally about where the locus for therapy should be, but every patient is different. So here we actually have the potential, if we can scale this, of personalizing these treatments for these debilitating diseases. Now, in a similar study from Eddie Chang's group at UCSF, they used similar kinds of long-term recording to identify biomarkers associated with poor mood states. They identified the area that they thought was optimal for deep brain stimulation, and using a two-way sensing actually deployed this in an adaptive state, in an adaptive paradigm where they're actually reading out neural activity biomarkers in real time and stimulating the patient back and showing that they can actually effect significant improvements in that patient's mood state. So this was the stuff, I think, of science fiction 5, maybe 10, maybe 5 years ago. And we're right now seeing the reality of being able to deploy these technologies, existing technologies, but in a different way, using very sophisticated machine learning algorithms to read out these signals to treat these devastating behavioral and mood disorders. So stay tuned, there's a lot more yet to come. These are results from early stage clinical trials, but now these trials are being expanded to see if they can be deployed more widely. Okay. So the second key takeaway I'd like to leave you with is that we are laying down the foundation for future discovery and future cures. Now, kind of continuing on this theme of neuropsychiatric disorders, to me it's sobering to realize that it's been about 5 decades since the introduction of serotonin-specific reuptake inhibitors like fluoxetine, Prozac, 50 years ago. And there has not really been a major drug developed since to address depression. Now, more recently, drugs like ketamine and various psychedelics have found an interesting resurgence in the application of these agents toward treating depression. So here's a study from the Olson and Tian labs at UC Davis that are seeking to understand the mechanistic underpinnings of how these psychedelics might actually be working. And if we have a better understanding of mechanism, we actually have a chance of designing better drugs 50 years after Prozac for treating these conditions. So here, the remarkable discovery they found was that it is the action of intracellular 5-HT2A receptors that appears to mediate plasticity, structural plasticity of these neurons. So here, what I'm showing is across the top of panel A are different variants of 5-hydroxytryptamine, okay? And as you add methyl groups, you increase the permeability of these agents into the cell. And what you can see on the bottom panel there in D is the increase in the dendritic complexity of these cells as you increase the permeability of these 5-HT2A receptor agonists that are now able to enter the cell. That's the idea. You can also increase the permeability of these agonists into the cell by overexpressing a serotonin transporter that literally just brings the drug, the agent, from the extracellular space inside the cell. So in the middle panel there is the dimethyl 5-methylhydroxytryptamine, which already is permeable, and you can already see increased bond density in both cases. But if you look on the right, when one applies 5-HT alone, the dendrite on the left looks pretty bare and naked, but when one increases the permeability of 5-HT into the cell on the right with this SERT, with this overexpression of the transporter, one can see an increase in spine density that's shown there on the lower right histogram. So that shows that the effects of 5-HT2A agonism on increased structural plasticity seems to be mediated by internal 5-HT receptors. And what they can also show now when they do this in vivo by injecting these agents into the medial prefrontal cortex is that it also seems to have some features that look to be antidepressant. So here we have a cell biological mechanism for how these agents might affect structural plasticity of the neurons themselves that apparently is associated with antidepressant activity. And it's these kinds of studies looking at mechanism that give us a pathway toward finding new therapeutics, new drugs for treating depression. So stay tuned. I think this will be interesting to follow in the next couple of years. Now, one of the recommendations of the Brain 2.0 report from back in 2019 was to invest in large projects that might really change the way we go about understanding how neural circuits work in the brain. And I would argue that in order to understand how this marvelous computer works, we need both a parts list for the brain as well as a wiring diagram, right? So there's 86 billion neurons plus about an equivalent number of non-neuronal cells that make trillions of connections with each other. What are their identities? How do they wire up to each other? And once we have that information, which is a required ground truth for this understanding, do we have a way of interrogating, asking questions about how these circuits function? So this gave rise to a project that we referred to as the Brain Initiative Cell Census Project, and the second phase of this project was referred to as the Brain Initiative Cell Census Network. So in full disclosure, I was a PI in this project before I joined the NIH 3 years ago, and our third speaker today, Dr. Xiaowei Zhuang, was also a PI on this project. And we undertook what started out as a pilot project, but wound up being a huge project to use a number of different modalities to characterize all the major cell types in the primary motor cortex across multiple mammalian species. This was published back in October 2021 as 17 papers in Nature, 10 other papers in Nature sister journals, and a whole smattering of papers coming after that. Just so this one little tiny piece of cortex, but it gave us some lessons about the organizational principles underlying diversification of cell types, of brain cell types. And it really also showed us the power of team science. We were using not just single-cell transcriptomics, but also epigenomics, spatial transcriptomics, physiology, anatomy, and connectivity, what have you. It involved over 250 researchers at over 45 institutions across 3 continents. So it really taught us a way of tackling these big resource-intensive projects by using team science in a way that I would argue had not been done in neuroscience before. It sets the stage for the Human Brain Cell Atlas, which is coming, which is now underway. The BICCN, or Brain Initiative Cell Census Network, is just wrapping up now. At last count, Xiaowei, I think there's about 90 papers in preparation that detail the characterization of the entire mouse brain as well as non-human primate brains and human brains as an initial marker. And already we're seeing from the tools developed in the Brain Initiative Cell Census Network, people are now applying these tools to identify cell types that are specifically vulnerable in Alzheimer's disease and Parkinson's disease in humans, not just models, but actually in humans. And this information will be critical for identifying future prevention and cures for these diseases. By knowing what cell types are affected, we actually have a shot at getting mechanism into disease initiation and progression, so stay tuned on that. Okay, so finally I'd like to talk a bit about how brain is creating a new paradigm for doing science that's accelerating the pace of discovery. So I mentioned the Brain 2.0 report. Back in 2021, 2022, we decided to invest heavily in 3 large projects, what we call the transformative projects, that we do believe will transform the way we conduct neuroscience research and be able to apply these resources toward finding cures for human brain disorders. The first is building on the Brain Initiative Cell Census Network, which initially focused on the mouse, but also started studies in non-human primate brains in humans. Now, in the current phase, the Brain Initiative Cell Atlas Network, or Access Network, or BICAN, our goal here is to map all the major cell types in the human brain, as well as non-human primate brains, and a little bit of cleanup work in the mouse. But here we're really shifting the effort onto the big brains, the human brains. The second project is this Brain Initiative Connectivity Across Scales, or Brain Connects project, whose goal is to develop the tools that will be necessary to develop whole brain connectivity maps in the mouse at synapse resolution, as well as projection level resolution maps in big brains, non-human primate brains and human brains. So these 2 projects together will give us the ground truth for what neural circuits look like. What are the parts? What are the different cell types, their properties, their connectivity? And what do the connectivity patterns look like at high resolution as these cells are communicating with each other in these complex neural circuits? And then finally, we need a way of interrogating this information, right? So basically, the Cell Atlas project, the connectivity projects will give us a set of testable hypotheses, and what we need now are more effective and scalable ways of targeting these cell types precisely to test their roles and the roles of the circuits that are involved in generating complex behaviors in animal models of both normal models as well as in models of disease, and ultimately to take this information on to treat human brain disorders, many of which are circuit disorders. Okay, so these are the 3 projects, and they are mutually reinforcing, and these are now underway. We've gotten generous funding from Congress to support these 3 projects. Right now, Congress has allocated specifically $135 million a year for these projects, and we're actually spending more on these projects because of the great response from the community. Now, I told you a bit about what's going on in the Cell Atlas projects. Let me just give you a taste of what's going on in the connectivity space and in the precision cell access space. So just to give you an idea of what might be coming in the future. So here's a paper that was recently published on the connectomic map at synapse resolution of an entire fly larval brain. Now, this was a study that was initiated through the support from Howard Hughes Medical Institute and the Wellcome Trust, and this was really performed at Cambridge and HHMI the Genelia Research Campus with some contributions from the Brain Initiative as well. So they've developed a full connectome at synapse resolution of the roughly 3,000 cells, neurons, that comprise the fly larval brain, along with about 500,000 synaptic connections, so really quite remarkable. They were able to categorize these neurons as either input, output, or interneurons, and through clustering, sophisticated clustering analysis, could group them into 93 types based on connectivity. Now, one of the really cool things is that they found that about 41% of these neurons showed highly recurrent connections. And many of those neurons actually were dopamine neurons that were in and around centers that are known to be involved in learning. So what people are using in advanced machine learning and artificial intelligence paradigms, nature has found a way to do it through evolution. And as the great Yogi Berra said, you can see a lot by just looking. So this is not only just a static map, but the actual nature of these connections helps tell us about how these circuits might be used in computation. So this is really cool. So 3,000 neurons, a mouse brain is roughly 80 million neurons, so we have about 4 orders of magnitude to scale up, and then another 1,000-fold to go up to humans. So you can see this will be a big lift, but what we're counting on is a development of technologies through these kind of projects that will accelerate the pace of discovery and bring these projects to fruition within the next 5 or 10 years. Okay. So very important, in order to be able to test hypotheses about how neural circuits function and how the cell types that comprise these circuits function, is the ability to access them. So we are also funding a set of projects, right now, six major projects, to develop gene delivery tools for accessing cell types in the brain, the postnatal brain. As it turns out, all 6 projects are involving adeno-associated virus as the key vector. So there's 2 ways to gain specificity into cells. One is by leveraging the information we're getting from the cell census projects to identify enhancer elements that will specifically express genes, payloads, whether they be optogenetic payloads or other kinds of sensors or gene editors, into specific cell types. But another very important aspect is getting these viruses into the right cells. So viral tropism is another strategy to gain specificity. Now, one very desirable aspect of doing these kinds of experiments, and ultimately in gene therapies, is the ability to inject these vectors into the bloodstream of the animal and have them cross the blood-brain barrier and into the brain, and not have to necessarily inject it directly into the brain itself. So here's one of several studies from Viviana Gradinaro's lab at Caltech, where they have used directed evolution to identify capsid protein variants that specifically target these viruses across the blood-brain barrier into the brain. So you can see in the top row is regular AAV. On the right-hand column is liver. That's where you generally don't want to be. So the garden variety AAV9 will go to the liver and not the brain. And here they show a series of variants that actually target the brain, which is in the left-hand column. So they have tropism to the brain, away from the liver. They've also identified another variant. You can't see these little things on the right are too small. Another variant that targets not only neurons, but also non-neuronal cells as well. So we want access to all the different cell types in the brain. So this is really one great step forward. And what's really cool is that second variant also targets, crosses the blood-brain barrier in marmosets, non-human primates, and targets these cells broadly in marmosets. So now we have a tool that's useful for targeting cell types in rodents, as well as in non-human primates. And recent work from the Gradinaro lab has now used a more directed approach to identify some of the receptors that are responsible for carrying these vectors across the blood-brain barrier, some of which are highly conserved across vertebrates. So there's really a great possibility here to develop these targeting vectors that will be good not only for basic research, but eventually for precision gene therapies in humans. So we're really looking forward to further developments in this space. Now, one interesting aspect of the Brain Initiative is we are funding research that takes advantage of the situation where patients that are being monitored in an epilepsy monitoring unit for future therapy or undergoing intraoperative recording have electrodes placed in the brain. And while they're waiting for these results for these monitoring studies to occur, some of these patients are willing to participate in actual research studies. So we have the opportunity to understand how neural circuits function in humans, which is really quite a remarkable opportunity that we are very interested in pursuing. Here's a study from a group at UCLA, which has developed a miniaturized monitoring and stimulation device called NeuroStack that allows them to record both single unit as well as local field potentials from patients that are implanted with stereo EEG leads awaiting for future therapy. So they're being monitored. But what's interesting, what's really cool here is that because of the miniaturization, the patient can actually get up and walk around the hospital room. So now there's a possibility of monitoring neural activity during somewhat naturalistic behaviors. I refer to this as studying awake behaving humans, okay? And the benefit here is that they can simultaneously monitor neural activity with great precision, as well as behavioral features, which will allow us not only to understand how neural circuits function, but because there's also the stimulation parameter as well, it can actually help design better closed-loop or adaptive stimulation protocols for patients, for example, suffering from Parkinson's or other movement disorders. So this really opens up a window to studying humans in naturalistic settings, and also sets the stage for devising better therapies for patients with epilepsy, Parkinson's disease, Alzheimer's, and what have you. Now, of course, this brings up some very important ethical concerns about working with humans, and I'll get to that in a moment. But this is a harbinger of a future project that we just launched at the Brain Initiative, which we call the Brain Behavior Quantification and Synchronization Program. And the goal here is to marry precision measurements in both time and space of neural activity together with better measurements of behavior to really start getting at the neural circuit basis of behavior. And we've just launched through two RFAs in the space of looking at human studies in a phased awards, as well as early studies to develop some of these programs in non-human organisms. And this is just launching it, and it will be expanding. I think this audience might be particularly interested in some of these projects, so please stay tuned for that. So what we're doing in the Brain Initiative has clear ethical, legal, societal implications, especially as it pertains to work with human subjects, not just in clinical trials, but in these other trials, other experiments that I just outlined for you. We take this very seriously. We need to look at the implications of what we're doing in a proactive way. We have a neuroethics working group that advises us very carefully about what we're doing, hopefully ahead of time before we get anywhere near being in trouble. We have a set of neuroethics guiding principles that include not only issues around safety, of course, and privacy, which are both paramount, but also ones of agency, ones around dual use and malign use, and really engaging the public and helping us understand what the important parameters are and what the benefits could be of these new technologies together with the associated risks. We hold a series of workshops on this subject. We have a team, a project team that actually looks at neuroethical issues as well as oversees funding of scholarly work in the neuroethics space. So this is very important. We really weave this into everything we do. Okay. Another aspect of the Brain Initiative is we have a deep commitment to open science, and we cannot have open science unless there's free sharing of the data and resources that are generated. Some of you might already be vexed by the new NIH's data management sharing policy. The Brain Initiative has been a bit ahead of this curve. Starting a few years ago, we are now requiring any brain project to deposit any data of import into one of our 8 brain data archives, and this is very important for making the data fair, accessible, interoperable, and reusable. Now, what we do is determined by who's doing it, and we hold very strongly that scientific excellence can be enhanced by improving the diversity, equity, and inclusion of our workforce. So we're very committed to building a stronger workforce. There's been a growing body of evidence in the research literature showing that diverse teams tend to be more creative and outperform teams with more linear or homogeneous viewpoints. My colleague, Dr. Michelle Jones-London, will give you a really nice overview of the various training programs we have for building a stronger workforce. We've also been running some pilot projects to bring in institutions from idea states as well as minority-serving institutions. And one innovation that Brain has undertaken over the last few years is this plan for enhancing diverse perspectives. So you're going to start seeing this in all the Brain RFAs coming out, except for the training awards. And it's also being rapidly adopted across NIH. So here the idea is we are requiring our applicants to include in the application itself an articulation of how diverse perspectives, it could be from different scientific disciplines, it could be from folks from different personal backgrounds, different career stages, different institutions, different locations in the country or around the world. How will those enhanced perspectives lead to a stronger project? And they need to summarize this in a one-page statement at the end, and this will be score driving. And we're already starting to see an effect where people are really taking into account how a diverse team can actually lead to better science. So stay tuned for this, and you will also start seeing this across NIH. It's already in about over 100 funding opportunity announcements. Okay, so I'll just wrap up here. I hope I've left you with these 3 takeaways about how we're already having an impact in the clinic, how we're laying the foundation for future cures, and how we're trying to create a new paradigm and a stronger workforce for accelerating the pace of discovery. And, well, there's a 4th takeaway. And that's the importance of continuing to build this momentum, and I hope that we can engage more and more of you in this effort. Just as a quick reminder, we have some upcoming events. We have a workshop in the Brain Behavior Quantification Synchronization Program looking at the opportunities in bringing in new kinds of sensors for measuring animal and human behavior. And then, very importantly, we have our annual Brain Initiative meeting coming in June. You do not have to have brain funding to attend. We welcome everybody who's interested in what we're doing to attend. It's a terrific meeting, and it will be held in hybrid format. So even if you don't want to drag yourself to Bethesda, you can participate virtually. You can stay up to date with what you're doing through these various avenues. If you don't mind, a few more emails in your inbox, and you can learn more about us at this website. So thanks very much for your attention.
Thank you, John, for giving me a nice segue into this. It's really interesting. When I woke up this morning with everything that's going on in the world, you have a lot of fears, anxiety. I never thought I'd be worried about a rubber chicken, but here we are. So today I will really try to, as briefly as I can, talk about not only the diversity training opportunities within the Brain Initiative space, but also in neuroscience in general, knowing that what I always like to do with any of my talks is that all of you can have a little goodie bag to take home, even if it could be for yourself, someone you know, you as a trainee, or also just you as a mentor. Hopefully you'll find something in this brief presentation useful in terms of taking back. So John already laid the stage in talking about the Brain Initiative, but I'm going to step a little bit broader and talk about neuroscience research at NIH. Neuroscience research, a lot of people, when we think about our investments and the bigger institutes, you think of NCI, you think of cancer. But what you need to realize is that across many institutes, the NIH spends billions to support neuroscience research. And sort of how we've thought about the Brain Initiative in terms of this collaborative effort, there also is a program called the Blueprint IC that takes on this theme of collaboration too, 14 ICs and centers. And under John's leadership, actually the Blueprint and the Brain are joining forces to do a lot of creative things within diversity training. So the Brain Initiative and the Blueprint work together under this umbrella of the Training, Inclusion, and Equity team. The leadership is housed under the Brain Initiative, but it includes some of these Blueprint programs, which is that collaborative effort of those 14 neuroscience ICs. And the goal really is to train a new and diverse generation of scientific leaders, because that's important. We talked a lot about this diversity of research, diversity of tools, but how science works, at the end of the day, it's about the people, and making sure that we have that heterogeneity to spur that innovation is so incredibly important. This slide is showing you, and I'll talk about some of these programs in detail, but not all of them. We've really tried to build a pathway, starting, in some cases at the very beginning, K through 12 programs. More recently, the Brain Initiative joined the Science Education Partnership Awards, and several neuroscience ICs separately, or I keep saying ICs, but institutes, separately are also part of this FOA. And this is really about providing that exposure and scientific literacy early on in the K through 12 program. As we go up the ladder, I'll talk about the Endure all the way to the supplements. So starting at the beginning of this training, exposure, and investment, we have the Endure program. This program originally started out about 11 years ago with these 14 institutes and centers, but then the Brain Initiative also has more recently joined on into it's so hard for me to say NOFO, but what we used to call FOA, that'll be coming out in a few months. And so the Endure program prepares undergraduates from diverse groups to enter and complete neuroscience PhD programs. And it's a program we're really proud of. So far, about 65% of the Endure trainees go on to PhD programs in neuroscience. And if you know, that's a long, if you know in terms of that investment at that undergraduate stage, it could go a lot of different ways because, their first step is just finishing up the BS or the BA, but 65% of them go on to do the PhDs. And the way this program works is by partnering them during their 2-year exposure with T32 programs or graduate neuroscience programs. And over those 2 years, these Endure students participate in, and we use this word very intentionally, authentic neuroscience research experiences, and really go through this career development and professional development so that by the time that they're graduating, they've had not only research experience during the school year, but most importantly, these full-time summer research exposures where they're getting to know the graduate programs that will hopefully end up recruiting them. And so far with our outcomes, we're seeing that that's true. So this is a program that if you're a PI, a T32 PI, a mentor, keep on your radar because the NOFO for this next cycle of funding will be coming up. Another program that's newer in our pathway of programs is the DSPAN or F99K00. Really excited to see that. I know that a few of our DSPAN are actually at this meeting right now. This program was created, so after we had made this investment in the Endure, we really thought about these people that we were sending into these PhD programs. Often, they were an N of 1, right? And as we know, along each step of the pathway, there is this rethinking about, do I belong? Do I have the self-efficacy to do this? Do I feel supported? And so creation of the F99K00 was sort of shoring up that next step of the pathway to really give the support, career development, but also the empowerment for these very, very talented and skilled trainees to see themselves in this neuroscience academic environment. And so the program provides 1 to 2 years for this F99 or graduate student to finish up their PhD defense. And then when it's time for them to go on to their postdoc, they have this golden ticket of 4 years of research funding. And we call it a golden ticket because the thought is that they are empowered to choose the lab and the mentorship that they really deserve, that they are coming with their own funding, and that the expectation is that they will be intellectual contributors to the research that's going to be proposed for their postdoc experience. And that has been really, we'd hoped that it would be a game changer, but hearing from our scholars, it really is. The other point of this program is it creates a cohort and a group of scholars. This morning, I think it's Bianca talked about the sense of competition and, people feeling like it's a Shark Tank. And with this community, I'm so happy to say it really is a community that people feel like they can share with each other anxieties, opportunities, learning moments, and just peer mentoring circles. And if you don't have to believe me, all you have to do is go to Twitter and follow the D-SPAN. It's a group that, me personally, it makes me jealous that I didn't have that when I was going through my own training. As we go up, the other program that we have in the Brain Initiative, and this one is open to all. The other few programs that I talked about really focus on making sure we're increasing underrepresented researchers, but this Brain Initiative F32 is open to everyone. Well, I shouldn't say everyone. The eligibility is that you have to be a PhD student that's, in their later stages, or a postdoc with no more than 12 months of postdoc training. And the thought here, as I talked about the D-SPAN, I talked about having this ownership and this intellectual contribution to a research project. And that's what the F32 does. You can identify a postdoc project, work with the mentor, even before you physically set foot on site, and come up with a project that we do not require any pilot data, right? We can't. You haven't even gotten there yet. So it's a really fantastic way for someone to creatively come into this Brain Initiative space. The other question that we get a lot of for the Brain Initiative is, suppose my graduate work really wasn't what you would call the Brain Initiative, but I know that for my postdoc, I want to move more into that field or that area of research. Is it going to be a problem that I don't have the papers or I'm just starting out? This F32 is a wonderful way and a wonderful mechanism to enter into that space. As we go up the career ladder now, so we've talked about the earlier postdocs, now we're talking about the advanced or later postdocs, and then also how do you transition people into their first faculty position. And so the Brain Initiative K99, Diversity K99 R00, in some aspects, it's very similar to the general K99 R00 in terms of the review criteria and what you would expect in terms of really dedicated mentorship, a solid training plan, and a plan for this transition. But the key features that make it different, and when we really were the first diversity K99 at NIH, was the fact for this one, we have an eligibility criteria of 5 years. And the thought behind that was that we knew for some of these groups, especially because this was a program, that we also wanted to include women in general, given the quantitative fields that we represent. You may say in neuroscience in general, women, yay, we're at parity, maybe a little bit above in terms of some graduate programs, and even in the postdoc, the numbers are looking equal. But for fields like engineering, computer science, mathematics, physics, it is not the same. It is really low. And these are people and fields that the Brain Initiative really wants to recruit to the research that it conducts. And so women are included in what we call our examples of diverse categories. And the 5 years was because we know if you are switching these fields, you may not have the perfect linear trajectory and be able to put in a competitive application in that 4-year cutoff. And so we were able to negotiate for a 5-year cutoff. And so the K99 would give you funding in the advanced part of your postdoc, funding for 1 to 2 years to finish that up. And then once you secure a tenure track position, you would be able to activate your R00. And then I'll talk about the research supplement program. So the Brain Initiative has a specific dedicated, what we call notice to the general diversity supplement program, meaning that we can fund it with Brain Initiative funds. But the diversity supplement is this rare thing that all NIH institutes and centers have. You will often hear when any of us are up here from NIH, we will always say, well, they don't have this, or this is how we use that, and this is. Now, we do use the diversity supplement a little bit differently at each IC, but this is one program that all of the NIH institutes and centers have. And the goal behind this program is if a PI has a grant, an R01, a U, a P, there's a whole alphabet list of eligible mechanisms, you can apply for an administrative supplement to bring in funding to fund a diverse trainee. And the trainee can be anyone that's, maybe in a high school program all the way to junior faculty. This is a win-win in terms of not only for the trainee, in terms of getting mentorship, pilot data, other things to then go on and launch and bridge into their own individual award, but it's a win for the PI in terms of, pushing forward the specific aims of a parent grant to now have this talented person helping you with that. And so on this slide, I just list not only the parent FOA, which is just the general FOA, but several of, some of these are congressionally mandated areas like ADRD, HEAL, that also have specific call outs for this diversity supplement program. And then this goes over, I talked about, each IC looks a little bit different in terms of, what they'll emphasize in terms of the review. The Brain Initiative, very similar to NINDS and NIMH, we really are looking for this to be a launching point for this candidate, that there should be evidence that the project, the experience is really going to set them up for success. And the thing that we'll look for in review is what is the transition? If you're taking in an undergraduate, what is the transition? Are you helping them? If you say they want to go on to graduate school, in that training plan, you should be talking about how you're helping them prepare for that. Are you, not all schools take GREs anymore, but, if that's the case and that's where they want to go, how are you helping them do that? How are you helping them to do all of these steps to get to that next transition? And real strong evidence that the person is receiving actual career mentoring. We don't want to fund anything that's a pair of hands. We want this to be a meaningful experience that leverages an opportunity for the person to go on to their next steps. The other K-99 that we have that a lot of neuroscience institutes participate in is the Mosaic program. And so this was a program started by NIGMS that many other institutes then signed on to. The key thing here and the difference here is that this program does have a DEI statement. So the applicant would talk about, their activities are, their sort of alignment with increasing DEI in science and research. The program, like all of the K-99s, provides up to 5 years of funding, but this one, the cutoff is 4 years. The other difference with this one is once you receive the K-99 R00 award, you would then be embedded as a cohort into one of these scientific societies that would then offer career development support. In terms of moving on to the faculty level, that transition there, NIH has made some really important investments, and one of them is the NIH FIRST program. Neuroscience has a really, I would say, broad presence within the FIRST. Many of the FIRST programs are looking to recruit neuroscientists. And the thing you need to know about the FIRST program is that the goals here are to really target the structures of an institution. So a lot of the programs I spoke about are individual awards, right? And these are awards to support an individual and to help them thrive and survive. But the reality is that, you know, we've come to the conclusion that sometimes we're not trying to fix broken people, right? We're putting people into broken systems. And especially for women and underrepresented groups, it's more about trying to change that culture and environment. And so that's part of the goal of the first program, is to really change these environments into inclusive research excellence environments, and at the same time provide funding to recruit scholars or faculty that have shown a commitment to DEIA. And so these are, these are programs that you should look out for. They have a very aggressive timeline in terms of trying to recruit junior faculty. And, you know, as I go around, I often hear junior faculty saying, like, nobody's recruiting. These people are recruiting. So take a look at these sites and see if your research aligns. The other, and I'll start with the problem first. And the other thing that we have sort of admired for a while, and I say that very sarcastically, is, the disparity in terms of the R01 data for underrepresented groups, especially African Americans, right? Many of you are probably familiar with what is known as the Ginther paper, showing, embarrassingly, the funding that NIH has towards these underrepresented groups. And knowing that, there's not one place along the system, but there are several places along the system where we're actively trying to address what may be these, things that come up with regards to bias. The data that are the little people that I'm showing you are actually our NINDS numbers, showing that not only, if you look at it, what you can see is that the percentage or the success rate for what we have coming in and what we have going out is not really that bad. But the numbers themselves are so woefully low. And so in 2022, we took about a very strategic, laser-pointed effort to solve that problem. And it started with 3 ICs, and now we're up to 9, although the ICs are listed at the top of the slide in terms of who are supporting this, what we call diversity R01, but it's an R01 that really is also for new and at-risk investigators. New is anyone who has never received a substantial independent funding from NIH. R21s don't count, and it also includes ESI. But the reality is that for early stage investigators, most ICs already pay early stage investigators points beyond the pay line. For example, at my institute, NINDS, say our pay line is 16th percentile, the ESI would get paid at the 26th. So it really is a program that hopefully will show bringing in people that are new investigators and also at-risk investigators, which are people who have not had a substantial independent award and will not have one in that current fiscal year. With the thought here that if we're building all of these pathways of our diversity training programs, we don't want to build a bridge to a cliff, right? We have to go that next step and make sure that people are retained into our biomedical workforce. I've already talked about that. So the next opportunities that I'll put on your radar are generated out of the Unite initiative. And so that is a new initiative from NIH with the pretty, I think, very bold goal of ending structural racism. And the Unite, each letter stands for a different part of the goals of the entire initiative. But the one that'll probably be most important to you is this E. And this is going to focus on extramural research, the extramural research ecosystem, changing policy, culture, and structure to promote workforce diversity. So coming out of the Unite, one of the more recent things, and I think this was released maybe a couple of weeks ago, is the Reward R01. So remember, I just told you about a diversity R01, but the Reward R01 is different because it has no ties to any demographics or eligibility based on being underrepresented in science. The Reward program provides support for scientists who are making significant contributions to DEI work. And, really maybe because of that effort and that investment, it's impacting their ability to do their science. And so with this R01, you can ask for both. You can ask for money for your research, but also your DEI efforts. And in terms of the review, they will actually judge not only your science, but also the impact you're making in the DEI space. And once again, there's no individual eligibility. This is open to all, and it will have standard R01 due dates. The other thing that's going on is the NIH Institutional Excellence in DEI in Biomedical and Behavioral Research Prize Competition. And so what's important here is that it's a prize competition. So anybody who feels as though they have a model program that really has identified practices for enhancing DEIA within faculty, postdoctoral scholars, student bodies, even grassroots efforts that may be put on by postdocs. They can apply for this through their institution, and it does not have to be tied to you actually having an existing NIH grant. You may have heard of the supplements, the DEI supplements, that you had to have an NIH grant. But this prize competition, it could really be anyone. And so NIH will award up to 10 prizes of $100K each through this competition. And half of the prizes will actually be set aside for consideration for limited resource institutions, because we know, sometimes those with the least maybe are doing the most, but then when it comes to that competition, it can kind of get washed out because of the magnitude. So this allows a level playing ground for all institutional types. And this slide, I'm just going to end with some of the, I've talked about a lot of the BRAIN programs, the blueprint programs. I wouldn't be able to go back to my office if I didn't show you, or my institute, if I didn't show you some of the wonderful work that NINDS is doing across the high school and the new faculty, really showing you that I think we talk about the training space, maybe with not always the optimism and the hope that we should be framing it. I think for trainees and coming into this space now, it's actually a pretty good time. There are several programs that not only can fund you at different career points, career development programs like the R25. Of course, every program isn't going to be for everybody, but we really do think that we have a very supportive pathway of programs, not only at the institute level, but within the BRAIN Initiative and also the Blueprint. It's important to, as you go about your journey in finding, what's the right program for me, it could be like a double-edged sword. Yay, there's so many programs, but man, there's so many programs. How do I figure all this out? The most important part to start with is what we're now calling the NOFO. You may have heard them before as the funding opportunity announcement. It's always, before you set out on any journey, confirm which institutes and centers are participating. look at the eligibility and the budget restrictions, understand the priorities. This is all going to be communicated in the NOFO. And then you can reach out, and we want you to reach out. It is so important. We do not want you to waste your time on a technicality. Like, you should never not be funded for a grant because you just didn't read that, they wanted this and you didn't do that. It should be based on the science, not a technical foul. And in terms of contacting a program official, we say even that word, we all have different names. Some institutes call it a program director, a health scientist administrator. Really, reach out to us. We are humans, so you might have to reach out again, but that's okay. You're not bothering anybody, and don't take it personal if you don't get a response on the first time out. In terms of navigating, like, who do I contact? I've already talked about, you should contact the person that's actually listed in that funding opportunity announcement or the NOFO. But it also, at NIH, we have the NIH Reporter Matchmaker. By show of hands, who has used that? Oh, not that many. You really want to check out the NIH Reporter Matchmaker, not only for this in terms of finding a program official, but it is a database of all of the the NIH funded grants. I think VA and NSF, you have a way to get to it, but maybe not through that exact link. But it's really important so you can see the landscape of your research. You could see collaborators, or as we've been talking about, you could see competitors. You can see where your research is being funded. And there's also information in there in terms of what study sections those applications that were funded went to. So it's really a good idea to know that in terms of the landscape of your specific aims and your science. And all you have to do is cut and paste a draft of your specific aims in the AI box, and it'll spit all that information out to you. It's really important to contact us early in the application process, and just know that one IC is or well, as we say, 27 ICs are really just, one IC. Like, that we all think differently about budgets, about priority areas, and so you may get sort of connected to different ICs, and that's okay, because the goal here is to find the right fit for you and to find the institute that will help you get to the best chance of success. The other thing is to really importantly follow up after you receive your summary statement. Especially, for some grant mechanisms, a not discussed, 50% of a study section is not discussed. You don't know if you were at the very top of the cutoff or the very bottom of the cutoff. Your summary statement should help you determine that, but also your program officer should be able to have a conversation and walk you through that summary statement. And another thing that I could not go back to my office if I did not promote this is our podcast. How many of you have listened to the podcast? No. I was going to say, Dr. Jaraza, I better see that hand up. So the podcast is run by two amazing people in my office, Dr. Marguerite Matthews and Dr. Lauren Ulrich. And this podcast is it's on season 4 now because of the success and just the helpful information that it has. It's something that you could take with you while you're running, while you're cooking. It really breaks down, season 1, it broke down the life cycle of a grant with the NINDS staff. So demystifying and giving you the behind the scenes sort of details of how we do what we do. The second season was the components of a grant with awardees. So people with boots on the ground telling you how they thought about and how much time it actually took to submit a successful application. And season 3 was all about mentoring, and some of these faces, I think are gonna be at the meeting today too, really engaging conversation about mentorship. We almost titled it The Good, the Bad, and the Ugly, but we thought that maybe that would be a little too inflammatory coming from the federal government. So, but it's a great, honest listen that yeah, you would be surprised how entertaining these podcasts are, is what I'll say. So take a listen if you can, and it's free. John has already showed this in terms of the Brain Initiative meeting, but I also want to put a plug in, since my talk is really focused on early career and trainees, the fact that at that meeting, if you do end up dragging yourself to Bethesda, is that on June 11th, from 5 to 8 o'clock, they're having a Brain, Neuroscience, and Beyond, building our early career community. And this is really going to be, I think, a really informative session that will focus on NIH grants, tips and tricks, peer mentoring, matching, and networking. So we would really love to see you there. I'll be there if that matters. And thank you. Thank you, everybody.
We have our final speaker, Dr. Zhuang. Please come to the podium. Thank you.
All right. So, well, thank you, Xiangmin, for the kind invitation. And I will actually have an entirely scientific talk, and because I didn't know I was supposed to talk about technology transfer, so that there's nothing about technology transfer in my talk. And then the other thing is, for some of my slides, I do think I need to point to things, and then I realized for people sitting there, seeing the screen with pointer might be difficult. So if you don't mind, and if you want to see the screen better, and then moving towards this direction, you know, that would be helpful. So, all right. So, actually, John had already sort of alluded to what I'm going to talk about. So what I want to share with you today is a new direction of technology that is spatially resolved or spatial genomics. And in our case, it's also single-cell resolution, so it's spatially resolved single-cell genomics, and with a focus on applying it to build the cell atlas of the brain. Okay, so let me start with some motivations. So, as we know that our body, and then also in particular our brain too, are made of many different types of cells, and these cells have different behaviors and functions and so on, in a large part because of their different gene expression profiles. So if you can do gene expression profiling of individual cells, then you can identify and discover cell types, and then understand the molecular basis of their different behaviors and functions. And indeed, single-cell RNA sequencing does just that, and then has already been transforming many areas of biology. So you might wonder, why do we want to develop, in our particular case, an imaging-based single-cell genomics approach? And the key word here, I mean, there's a number of advantages, but one key aspect is the spatial information. Think about if we could use imaging and then in intact tissues without dissociating the cells, and then profile the gene expression of individual cells, so then we can identify and discover cell types directly in situ, and then get this spatial organization. Or I should say that the spatial organization in many cases, and of course in the brain too, is really critical for brain function or tissue function. So by doing in situ gene expression profiling, identifying cell types in situ, we can directly probe this functionally important spatial organization. All right, And there's more things. For example, the spatial information is not just important at the cell organization of the tissue level, but also intracellular level. We know that transcripts are not randomly distributed inside cells, and the intracellular distribution of RNAs are actually an important post-transcriptional regulation mechanism. So spatially resolved transcriptomics could also get us this intracellular transcriptome organization. And likewise, if we apply it to DNA, and we also know that our DNA as a polymer is not randomly folded, and the 3D organization of the genome is also an important gene expression regulation mechanism. And then, of course, important for gene expression regulation is the epigenomic properties, which if we can actually get the spatially resolved information or spatial information, that's also very important. So how do we actually, so we do that by imaging. So in other words, we do genome scale imaging. So how do we approach or how do we achieve genome scale imaging? And there are multiple approaches to do that, and the approach that we developed is called MERFISH. And so the nutshell is these three key ideas, and that is error-robust barcoding, combinatorial labeling, and sequential imaging. So specifically, if I use RNA as an example, what we do is we assign unique barcodes to individual genes or to different barcodes to different genes. For example, in this case, these binary barcodes. And then we physically imprint the barcodes onto the RNAs through combinatorial labeling with a high diversity of our library of encoding probes, and these probes can not only recognize specifically RNA molecules, but also have these readout sequences that corresponds to their barcode. For example, if an RNA has a barcode of, I couldn't even read it myself. So let me try this. I'm not positive I can do this, but let me try this. Sorry about that. So if we have a barcode of 101 and so on, so an n-bit binary barcode, so for n-bit binary barcodes, we basically just have n different readout sequences. And if we have a barcode of a 101 and so on, then for this RNA, it will have readout sequence 1, but not 2, readout sequence 3, but not 4, and then eventually readout sequence n. And then we will detect the presence of these readout sequences by a sequential round of single molecule FISH measurements or hybridization, where the readout probes are complementary to the readout sequences, so that we can determine what are the readout sequences that are present on the RNAs and enhance its barcode. And in these individual rounds, we use single molecule FISH, as I mentioned, which is a technology or method pioneered by Rob Singer and advanced by Singer, Ajahn Raj, and others into a really highly accurate approach. So, and as I said, to achieve genome scale imaging, the key things that we did, invented is the combinatorial imaging, labeling, and sequential imaging. And the key advantage of that is its scaling capability. And you can easily imagine that if I have n-bit imaging, then I can distinguish 2 to the n different kinds of genes, which means that we can use a relatively small number of rounds of imaging to get a very large number of genes. You know, for 16-bit, you can do 16 rounds of imaging, or you can even do 4-color imaging with only 4 rounds, okay? So that's an advantage, but in order to do this multi-bit imaging, it also comes with a significant challenge, and that's error accumulation. I already mentioned that single molecule FISH is very accurate, only a few % error, but that's per bit. So after n bits accumulate to a very substantial misidentification rate, and we overcome that with this error-robust barcoding, which can detect and correct error, and then hence allow us to get high accuracy measurement and comparable to, if not higher than, single molecule FISH itself. So I won't have time to go into really the details of this part, but the paper was published a long time ago, so if you're interested, you can go there for details. So that's why we call our approach multiplexed error-robust FISH or MERFISH. And I also won't even have time to show the data in this paper. I'll just show some more recent one. This is just because the author of this paper, my postdoc made this really beautiful image. So that really allows you to see visually that individual dots corresponds to individual RNA molecules. This is the image of actually more than 10,000 genes, 10,050 genes. The individual dots are individual RNA molecules. Their color actually is sort of reflected the genetic identity. So this actually itself already shows you that there's indeed interesting intracellular spatial distribution of RNA, which I won't be able to cover today. And then I will just quickly mention that this method can be applied to also looking into DNA and the 3D organization of the genome. And the concept, I won't do too much about the assay development description, but just to give you a concept, instead of using similar to the case of RNA, instead of using the MERFISH to identify RNA genetic identity, we use that to identify genomic loci in the DNA polymer, and then we can actually identify these genomic loci and then trace their 3D organization. And we can actually do multimodal genome scale imaging of not only DNA to get the 3D genome organization, but also genomic scale imaging of RNA simultaneously so that we could look into the correlation or relationship between the 3D genome organization and the transcriptional activity. And then we can also do protein imaging simultaneously in the same cells, such as in this case, we image these landmark nuclear structures so that we can understand the 3D genome organization in its functional activity context and the nuclear structure context. And then recently, we also extend this to epigenomic imaging to actually image epigenomic properties such as histone modifications at the genome scale. And what we do here is we use antibodies to recognize for example, histone modifications, and then bind protein A linked to transposase TM5 to the antibody. And then the transposase will cut the DNA and attach specific sequences at the end in a way similar to the cut and tag approach developed by Stephen Hanakoff's lab. And then the tag that we put on the end of the DNA is actually T7 promoter, which can amplify that into multiple copies of RNA we then detect with MERFISH. And then just to show a couple of sort of results panels. So for example, we use this to image a large number of putative enhancer elements. And then actually by looking at the enhancer spatial distribution and a nearby gene spatial distribution, and then if they are strongly correlated, we can even use that to predict potential promoter enhancer pairs. And with that, we also observed a very sort of frequently appearing phenomena is that the enhancer element or putative enhancer element potentially form these hubs. And these are 10 nearby enhancer elements that are all near this transcription factor gene. And then you can see that they have a spatial distribution concentrated in the dicephalon region that is very similar to the expression of this nearby transcription factor. Actually, all these things are within the same TAT. And then this transcription factor, TCF7L2, suggesting that these actually might form a hub to actually regulate the gene expression of this important developmentally important transcription factor genes. And interestingly, 7 of the 10 elements actually have a binding motif of a T7, have the TCF7L2 binding motif, suggesting that this transcription factor might actually bind to its own enhancer to form a feedback loop to ensure a robust transcription of this important transcription factor during development. So with that as a brief introduction of the technology, I want to now really just focus on our application of this, especially the RNA MERFISH part, to study the cellular organization or the molecular and cellular architecture of the brain. And we started with study various brain regions, which I won't have time to cover today at all. But I do want to mention that among these is indeed this primary motor cortex study that we participated in as this BICCN mini atlas that was published in that issue of Nature, which I won't be able to talk about that work today. But I want to actually focus on our recent work, and thank John for already mentioned that, indeed, of the whole mouse brain. And then I'll very briefly talk about a recent brain aging study, but the end was also very fittingly about this new BICAN project to show that we can indeed use merfish to study the human brain, okay? So with that, let me just jump into the whole brain study, which the preprint is already on bioRxiv. And then in this study, we imaged, we imaged more than 1,100 genes. And these genes are selected based on the single-cell RNA sequencing data that were acquired as a parallel study by our collaborator Hanghui Zhen's lab at Allen Institute. And these are the differentially expressed genes that mark specific transcriptionally distinct clusters seen in the single-cell RNA sequencing data from Hanghui and Zizhen and the Allen team. And then we imaged more than 8 million, we imaged about 8 million cells. It's really, I do want to applaud my postdocs for doing that. So it's like about 250 brain slices, coronal slices, sagittal slices, and so on, to see actually the cell atlas of the whole mouse brain. And then we integrated the merfish data with the Allen Institute single-cell RNA sequencing data, and with that identified a very large number of transcriptionally, actually more than 5,000 transcriptionally distinct cell clusters that belongs to about 300 subclasses of cells. And then, of course, because of the spatial capability of merfish, then we can see the spatial map. And this is just a very small subset of these, 250 brain slices laid out here. You can see the coronal slices and sagittal slices, and you can see this really beautiful, mesmerizing spatial map of these cells. And these are actually at the 300 subclass level, and then we can also plot it out at the 5,000 transcriptionally distinct cell cluster level. And one thing that's really quite remarkable is that it not only the cells have this extraordinary molecular diversity, it also has pretty much an equal level of spatial heterogeneity. Most of the transcriptionally distinct cell clusters actually take distinct spatial organizations or spatial distributions. And then this integration also has an added advantage, and that is it allows us to impute the gene. We imaged more than 1,100 genes, but this integration allows us to impute the genome-wide expression profile of every merfish imaged genes, so that we actually have the transcriptome-wide profiling too, which comes in handy for some of our later analysis. So I'll just give you some findings. For example, neurons, they're really extraordinarily diverse, and then we've seen even for neurons already more than 5,000 transcriptionally distinct cell clusters belonging to 283 subclasses. And then there are a lot of interesting spatial distribution stories that I won't go into details, but here in this slide, I will just talk about the neurotransmitter usage. For example, you can see there's a large number of high diversity of glutamatergic neurons, their spatial organization, a high diversity of GABAergic neuron, their distribution, as well as, these modulatory neurotransmitters, serotonin, dopamine, and so on, their spatial distribution. And not only neurons, but also non-neuronal cells also have a high diversity, of course, not as high as neurons, and we observed about 100 transcriptionally distinct cell clusters that belong to 23 subclasses, and then they also show very interesting spatial organization. And one particular type, astrocytes, is very pronounced, that it's really quite remarkable that the 24 clusters of astrocytes, each individual one cluster takes a distinct, unique spatial distribution. And functionally, what's the implication of that is, of course, open to discussion. So there are, a number of things that we can learn from the spatial organization, which I won't have time to go into in this talk, all of them, but I just want to highlight one of this is actually the high spatial resolution actually allow us to infer cell-cell interactions. And this we do by looking into the soma contact or proximity that occurs at a frequency that's higher than random chance. And of course, because we look into the soma spatial relationship, that means these interactions arising from soma contact or paracrine signaling, or maybe a short-range interaction of interneurons through their processes and so on, but it doesn't cover the long-range synaptic interactions between neurons, okay? So what we do is we look into, for example, any particular pair of cell types, what's the frequency of two cells coming into contact or proximity, and then locally permutate or spatially permutate locally the cell positions, and then see what's the random chance. And we do that locally just because, as you know, brain has an organization. We don't want to just capture the layered organization of cortex, for example. And then when we compare to that permutation results, we see whether this proximity happens statistically significantly. And with that, we identify interactions or communications between several hundred pairs of cell types. That's at the subclass level. Okay, and then more than that, because we have a transcriptome-wide expression information of every individual cell, we could also see whether certain ligand receptor pairs were upregulated in the interacting or proximal cell pairs, or other genes are upregulated. So that actually gives us a molecular basis of these interactions or even functional implications, as I will allude to in a few examples. So we've observed potential interactions. This I emphasize, these are a hypothesis of interactions. Of course, they actually, whether they are bona fide interactions, require additional validation. So we observed interactions between, among non-neuronal cells. For example, one interesting class is between vascular cells and immune cells. And here you can see statistically significant interactions between endothelial cells and this type of macrophage, border associate macrophage or parasites, and the border associate macrophage. And as I said, we can look into ligand receptor pairs that are potentially important for the interaction. And then for both cases, we identify the most pronounced pathway is the lambing pathway. And it's actually known previously that the endothelial basal membrane lambing actually is important for conversion or differentiation of monocyte into macrophage. So maybe this interaction is actually important for controlling the macrophage pool in the brain. And we can also look into other genes that are upregulated in these interacting cell pairs. For example, cytokines we found are upregulated in the endothelial cells or parasites that are interacting with the macrophage. And the cytokines are known to be chemoattractant of macrophages, so this might be another mechanism of actually recruiting macrophages to the brain. And then we can not only see interactions between these vascular cells and macrophages, border associate macrophages, but also microglia. And then for both endothelial and parasites, they show significant interaction. And also, interestingly, we found that for interaction with microglia, parasites are more preferentially interacting with microglia than endothelial cells. But it's the opposite for the border associate macrophages, and that is endothelial cells is more preferentially interacting with macrophages compared to parasites. And biologically, what's the implication of that is open to future investigation. And we can not only see interactions among non-neuronal cells, but also between neurons and non-neuronal cells. For example, a prominent example is this astrocytes immature inhibitory neuron interaction in the olfactory bulb. And then there we also see that among the upregulated ligand receptor pathways, Wnt signaling is one of those. And then if you look at the genes that are upregulated, there's one very pronounced upregulation of an interesting gene, SFRP1, which SFRP1's function in this case is unknown, but in a different context, SFRP1 in oligodendrocyte precursor cells is actually known to inhibit proliferation of neural stem cells. So suggests the possibility that astrocytes might use these genes to actually regulate the neurogenesis in the olfactory bulb through inhibitory neurons, and of course up to future validation of this hypothesis. And we can also see interesting neuron-neuron interactions. I might be actually behind on, so let me actually skip that part. There are quite a number of those, so I won't go into details of this. But just to actually summarize just this whole mouse brain study, and again, it's a very large-scale study where we imaged 1,100 genes in 8 million cells, many slices, integrated with the single-cell RNA sequencing data to build this whole mouse brain cell atlas, which we think will be a very useful resource for the neuroscience community. And we already learned from this that there is tremendous molecular diversity and heterogeneous spatial organization of not only neurons, but also non-neuronal cells. And we also identified the molecularly defined brain regions and the spatial gradients that I didn't have time to talk into, but that get into the details. I did actually give some examples of the cell-cell interactions, but this approach allowed us to really systematically to do a brain-wide investigation of cell-cell interactions, which actually give us hundreds of hypotheses of interacting cell type pairs, and the analysis of the upregulated genes, ligand receptors or other genes, give us a hypothesis of the molecular mechanism underlying these interactions and functional implications that can be tested. So with that, I just want to very quickly mention that as an imaging approach, it also gives you the ability to do multimodal imaging, because ultimately, once you see these different types of cells or different types of neurons and so on, you want to know what their functional roles are. So you can combine MERFISH imaging with functional imaging, such as neuronal activity imaging by calcium or voltage indicator and so on, and with that to learn activity or functional roles of these neurons. And I want to actually just cite one of our, or describe very briefly one of our very early brain region study, which is the hypothalamic brain region, preoptic region, to show that in this case we just actually simply included immediate early genes in the MERFISH panel to see after behavior such as parenting, which cells are activated by looking into immediate early gene being turned on, such as CFOS, and then identify the cell types. And with that learned, actually did a pretty broad survey of a number of social behaviors, parenting, aggression, and mating, and to see what are the different types of cells that are activated by each individual behavior. For example, if we look at parenting and see when we expose virgin male to pups, which is known to elicit parenting behavior, we can see that among some 70 subtypes of neurons in this preoptic region, there's one particular subtype, an inhibitory subtype, that is very pronouncedly activated compared to other cell types, which we call this parenting type, and that is also activated in mothers and fathers, and indeed, mothers and fathers, after exposing to pups, do also exhibit parenting behavior. But very interestingly, you can see that the same parenting cell type was not activated when virgin male was exposed to pups. And virgin male exposed to pups actually is known not to elicit parenting behavior, but elicit aggression or even infanticidal behavior. So this observation actually gives us and indeed, we see that it's a different cell type that was activated in virgin male exposed to pups. That was the same cell type was also activated when virgin male was exposed to adult male, which elicits aggression behavior. So this gives you the cellular basis to understand why virgin female mice exhibit parenting behavior, and so are mothers and fathers, but not virgin male mice. And then I won't go into details of these other things, but just to say that this kind of approach really help us to understand the genetically encoded circuits comprising distinct cell types that control specific social behaviors. And I want to actually probably just summarize this slide without going into too much details, that recently we applied that to study. Maybe, Xiangmin, you can tell me how bad I'm doing with timing. 3 minutes. Okay, let me just very briefly summarize this to say that when we applied to look into the cortex and the striatum of the mouse, and then we learned that aging induced more pronounced changes in non-neuronal cells as compared to neurons, and in particular, they show upregulation of genes related to inflammatory and immune response. And because of the spatial capability, we also learned that the corpus callosum was a hotspot for aging-induced inflammation, or activation of astrocytes and microglia that were correlated, potentially caused by oligodendrocyte inflammation. So that's just that. But let me actually spend the last few minutes talking about the human cortex study, which we're very excited about. And this is a study that we demonstrated transcriptome imaging by MERFISH of a human brain, and we actually imaged a very large number of genes, 4,000 genes. And because we started this as a technology demonstration work to show that indeed we can get to a very large scale, even in the more challenging human context where the autofluorescence background, for example, is high. And then we use that to study a couple of human cortical regions, the middle and the superior temporal gyrus, or MTG and STG. And this is a nice collaboration with also the Allen Institute, in particular at Lane's team. And then with that, we observed, again, a high diversity of cells, and remarkably, not only the neurons, but also non-neuronal cells shows a very high level of diversity, dozens of transcriptionally distinct non-neuronal clusters. And we compared human MTG and STG with a variety of cortical regions that we imaged in the mouse. For example, the motor cortex, as I had already mentioned, but also auditory cortex, because STG contains auditory cortex, and then also TEA region of the brain, mouse brain, which is the closest analog to the human MTG. And then you can immediately see that there is a very big pronounced difference in cell type composition between human and mouse, in terms that the human cortex has 5 times higher glial to neuron ratio, 3 times higher inhibitory to excitatory neuronal ratio, and 5 times higher intratelocephalic to non-intratelocephalic neuronal ratio. So here I won't talk too much, but just to say that not only excitatory neurons, but also inhibitory neurons and even some non-neuronal cells shows laminar organization in the human cortex. But end with this, I already mentioned how we infer cell type specific cell-cell interactions, and we observed something really interesting and also different between cell-cell interactions in human cortex and mouse cortex. And the methodology is the same as I had already explained for the mouse whole brain study. In fact, the Rongxing Fang, who led the study, developed this approach that we later on used for the whole mouse brain. So let me actually explain this more complicated plot, but the message I'll get to is very simple. And that is here each circle corresponds to the enrichment for interactions compared to random chance for a couple of cell types, for example, microglia cell and L23IT. And the circle size is statistical significance, and the color is the fold change. And then immediately you can see the difference between human and mouse. And then there are many interactions that are enhanced in human as compared to mouse. And I won't have time to go through all of them, but just to focus on one, and that is this group of interaction between non-neuronal cells, microglia, oligodendrocytes, and oligodendrocyte precursor cells with a bunch of neurons. That's much more enhanced compared to human. But I want to emphasize this enrichment part because that doesn't mean that these interactions are not there for mouse. It's just they're much more enhanced in human as compared to mouse. And just to elaborate a little bit, this is a group of interactions between oligodendrocytes and various neurons. And you can visually even see that these cell bodies of oligodendrocytes are neurons are often surrounded by multiple oligodendrocytes, and this multi-way interaction is enriched in human much more than in mice. And functionally, oligodendrocytes is known to be important for metabolic support of neurons. These are paraneuronal oligodendrocytes. And then also we know that firing individual neurons in the human brain requires more energy. So this might actually be an evolutionary adaptation that actually is important for sort of this higher energy demand. Oops, what did I do? Higher energy demand of firing individual neurons in human brain. Of course, paraneuronal oligodendrocytes also are hypothesized to have other functions. So it could be important for those too. So, and then let me just end with this one final thing that you can see that the microglia interaction with neurons are also substantially enhanced in human brain as compared to mouse brain. And microglia is known to be important for homeostasis of the brain, tissue homeostasis. So that might be indicating that this kind of a function is enhanced in human brain as compared to mouse. And then the ligand receptor pairs that we identify to be upregulated in the microglia and neuron interaction, several of them are known to be genetically associated with neurodegenerative diseases, actually suggesting the relationship between this interaction and neurodegenerative diseases. So with that, let me just end by saying that I hope I've convinced you that this approach of spatial genomics can allow us to get to molecularly defined and spatially resolved brain cell atlas with functional and cell-cell interaction annotation. And at the beginning part, I showed that we can also use MERFISH to actually get epigenome and 3D genome. So hopefully with this, we can even mechanistically understand transcriptional regulation in different types of cells and understand how they impact the development function and dysfunction of the brain. So with that, let me just get to the last slide of thanking the people who has done the work. And given the time, I will actually skip the apologizing to my students who developed MERFISH, including DNA and epigenomic MERFISH, just to only recognize the people who has done the brain work, in particular, Meng Zhang, Xinjie Pan, and Wang Zhang for leading the whole brain study, and that's a very nice collaboration with Heng Huizhen's team, and Rongxing Fan and Chenglong Xia for leading the human brain study in a collaboration with Ed Lane. And I didn't talk about hypothalamus work, so but that's a very nice collaboration. I did talk a little bit about it, and that's a very nice collaboration with Catherine Dulac, and the brain aging is led by Will Allen, who is a joint postdoc between Catherine and I. The work is supported by Hughes and NIH primarily, but also the early part of human study by CZI. And here is my one sentence about technology transfer. Actually, this is a financial conflict of interest disclosure. I'm a co-founder and consultant of Visgen that commercialized the MERFISH technology. So thank you very much for your attention.
So I guess, it's very exciting. Thank you, all speakers, for informative, insightful presentations. And this, I guess, because we have the next one coming soon, so I will encourage you, the audience, to come here to talk to our speakers if you have some questions, and also we'll follow up on more in the reception. Okay, thank you. Let's give another applause to our speakers. Yeah.